Hu.MAP 2.0, integration of over 15,000 proteomic experiments builds a global compendium of human multiprotein assemblies. Kevin Drew, corresponding author 1, 2 John B Wallingford, 1 and Edward M Marcottecorresponding author 1. Author information article notes copyright and license information disclaimer. Associated data. Supplementary materials. Data availability statement. Go-to. Abstract. A general principle of biology is the self-assembly of proteins into functional complexes. Characterizing their composition is, therefore, required for our understanding of cellular functions. Unfortunately, we lack knowledge of the comprehensive set of identities of protein complexes in human cells. To address this gap, we developed a machine learning framework to identify protein complexes in over 15,000 mass spectrometry experiments, which resulted in the identification of nearly 7,000 physical assemblies. We show our resource, Hu.MAP 2.0, is more accurate and comprehensive than previous state-of-the-art high-throughput protein complex resources and gives rise to many new hypotheses, including for 274 completely uncharacterized proteins. Further, we identify 253 promiscuous proteins that participate in multiple complexes, pointing to possible moonlighting roles. We have made Hu.MAP 2.0 easily searchable in a web interface, http://umap2.proteincomplexes.org/, which will be a valuable resource for researchers across a broad range of interests including systems biology, structural biology, and molecular explanations of disease. Keywords: data integration, human protein complexes, mass spectrometry, moonlighting proteins. Subject categories, methods and resources, post-translational modifications, proteolysis, and proteomics. Go-to. Abstract. Integration of orthogonal high-throughput mass spectrometry datasets using a machine learning framework reveals nearly 7,000 human protein assemblies and offers insights into novel protein function. An external file that holds a picture, illustration, etc. Object name is MSB17E10016G002.jpg. Go-to. Introduction. Macromolecular protein complexes carry out a wide variety of functions in the cell, including essential functions such as replication, transcription, translation, and protein degradation, for example, MCM or C, RNA polymerase, ribosome, proteasome. Alberts, 1998, Hartwell et al., 1999. The disruption of protein complexes is implicated in many human diseases, Go et al., 2007, Bruhl et al., 2017, and many therapeutics target protein complexes, Arkin et al., 2014. The formation of protein complexes to carry out biological function is a general principle of biology, and characterizing their composition is therefore a basic requirement to our full understanding of cellular functions. Unfortunately, we still lack knowledge of a comprehensive set of protein complexes for the human cell. To address this gap in knowledge, there is an ongoing worldwide effort to identify all protein interactions in human cells. High-throughput protein interaction screens using affinity purification, ATMS, Malavani et al., 2011, Hein et al., 2015, Hutlin et al., 2015, 2017, have greatly increased coverage of protein interactions across the proteome. Likewise, biochemical separation strategies, such as cofractionation mass spectrometry, CFMS, have provided orthogonal approaches to identifying protein complexes, Havujamana et al., 2012, Christensen et al., 2012, Kirkwood et al., 2013, Wen et al., 2015. Although these methods have identified tens of thousands of protein interactions, they still have limited coverage of the entire human interactome. Fortunately, these high-throughput methods are orthogonal, each sampling different parts of the human proteome and identifying sets of interactions which do not completely overlap. We previously reanalyzed and integrated 3 of the largest datasets available at the time, Hein et al., 2015, Hutlin et al., 2015, Wen et al., 2015, over 9,000 mass spectrometry experiments, to build a more complete and accurate set of protein complexes, Drew et al., 2017. Our resource, Hu.MAP, identified interactions for over a third of all human proteins. We envision interactomes as evolving entities, growing, and improving as new technologies and datasets emerge. Here, we introduce Hu.MAP 2.0, which we find to be the most accurate and comprehensive human protein complex map available to date. Hu.MAP 2.0 is an integration of over 15,000 mass spectrometry experiments and identifies 6,965 complexes consisting of 57,148 unique interactions among 9,963 human proteins. Multiple performance metrics demonstrate that Hu.MAP 2.0 outperforms our previous map and other complex maps available in the literature. We further show that complexes in Hu.MAP 2.0 are not only highly enriched for specific literature-curated annotations but also have greater coverage of completely uncharacterized genes. Finally, we highlight several new biological findings that illustrate the utility of Hu.MAP 2.0 as a resource for biological discovery. Go to Results. Our strategy in building a comprehensive map of protein complexes involves the integration of the many orthogonal experimental protein interaction datasets available using a custom machine learning pipeline as shown in Fig 1a. Each individual experimental dataset identifies different sets of protein interactions and therefore combining them results in a more accurate and comprehensive set of interactions. Our pipeline combines quantified features from these datasets using a support vector machine, SVM, classifier which calculates a confidence score of two proteins interacting. This results in a large protein interaction network. The network is subsequently searched for dense regions of highly connected proteins which represent individual complexes. The identified complexes are ranked by a clustering confidence value. We call the resulting set of complexes Hu.MAP 2.0. The map of complexes contains many known complexes such as the IF2B complex, spliceosome, RNA Pol 3, and IFTA complex, Fig 1a positive control examples, as well as many novel insights into the physical biology of the cell. An external file that holds a picture, illustration, etc. object name is MSP17E10016G003.jpg. Figure 1. Machine learning framework to identify protein complexes. Graphical description of computational pipeline to integrate greater than 15,000 mass spectrometry experiments. Number of experiments used is listed next to each technique, see also Table 1. A support vector machine, SVM, classifier was trained using numerical measures, i.e., features, on pairs of proteins calculated from original mass spectrometry data and training labels from literature-curated complexes, quorum. The classifier was then used to construct a protein interaction network by calculating a confidence score for all pairs of proteins for their propensity to interact. Clustering parameters were then learned from training complexes, and 5 final sets of clusters were chosen ranked in order of confidence from extremely high to medium. The union of these selected clusterings represents the final set of Hu.MAP 2.0 complexes. Networks of previously known protein complexes identified by this pipeline which were not in the training set of complexes are shown as positive control examples. Upset plot, Lex et al., 2014, displaying the intersections of protein pairs for all integrated datasets. Each set of connected black dots represents the intersection of the respective datasets. Vertical bar plot displays protein pair count of intersection. Light gray dots are datasets not included in the intersection. Single unconnected black dots represent protein pairs that are only present in a single dataset. Horizontal bar plot represents total protein pair count in each dataset. The plot shows the weighted matrix model. Single black WMM dot provides additional information for many pairs of proteins, greater than 2.2 times 10^6, that would be limited otherwise. Integration of over 15,000 mass spectrometry experiments. To construct Hu.MAP 2.0, we integrated over 15,000 previously published mass spectrometry experiments using our custom machine learning framework. We built upon the 9,000 mass spectrometry experiments used for Hu.MAP 1.0, Hein et al., 2015, Hutlin et al., 2015, Wen et al., 2015, Drew et al., 2017, by incorporating additional affinity purification data from Bioplex 2, Hutlin et al., 2017, and Bolt et al., 2016, as well as proximity labeling data from Gupta et al., 2015, and Yoon et al., 2018, Fig 1a. Our rationale for including these datasets was twofold. First, each dataset samples a different set of bait proteins which provides increased coverage of the interactome. Second, the methods are orthogonal and complementary, where affinity purification targets stable interactions, and the proximity labeling datasets potentially also capture transient in vivo interactions. The upset plot, Lex et al., 2014, in Fig 1b shows that tens of thousands of protein pairs are represented by two or more datasets providing orthogonal evidence for those interactions. This greatly enhances our framework's ability to identify true interactions from false ones. Additionally, we applied our weighted matrix model, WMM, technique which we previously demonstrated identifies many new high-confidence interactions from affinity purification data, Hart et al., 2007, Drew et al., 2017. Contrary to the traditional spoke and matrix models used to interpret APMS data that only consider one pulled-down experiment at a time, the WMM technique takes all pulled-down experiments from a given dataset into account and determines protein pairs that are in the same pulled-down experiments more often than random chance. The WMM balances both the false negative and false positive issues that face both the spoke and matrix models and therefore is capable of identifying novel interactions. More specifically, since a spoke model only considers interactions between a bait protein and a prey protein, all true interactions between prey proteins are misleading to high false negative rates for the spoke model. Alternatively, a naive matrix model does consider interactions between prey proteins limiting false negatives but does so by treating all prey pairs equally. Some of these prey pairs will participate in the same complex, but since proteins participate in multiple complexes, two prey proteins pulled down by the same bait are not guaranteed to interact. This leads to a high degree of false positives for the naive matrix model. The WMM considers all prey pairs as interactors but weights them according to the frequency they occur together while controlling for frequent flyer or sticky proteins. Therefore, by considering all prey pairs, the WMM has better false negative rates than the spoke model, and by accurately measuring the specificity of the prey pairs, the WMM has better false positive rates than the naive matrix model. The WMM can be applied to proximity labeling experiments and also, in contrast to the traditional models, the WMM can be applied to datasets that were not exclusively collected for the purpose of identifying protein interactions. Specifically, we applied our WMM to greater than 3,000 RNA hairpin pulled-down experiments, Triber et al., 2017, and incorporated the results into our framework. Figure 1b shows WMM overlaps substantially with other methods but also provides additional information for many pairs of proteins not covered in the other datasets, greater than 2.2 times 10^6 protein pairs attributed to WMM alone. Hu.MAP 2.0 network is highly accurate. We next trained an SVM classifier to determine whether two proteins interact in a macromolecular complex. The classifier uses 292 features computed from the mass spectrometry experiments and is trained on examples consisting of co-complex protein interactions from a set of greater than 1,100 literature-curated quorum complexes, Giraju et al., 2019. All features, with the exception of WMM features, were downloaded from the original publications. See materials and methods for a complete list. The set of quorum complexes were split into two equal proportion non-overlapping test and train subsets. We then derived sets of co-complex interactions from these test and train complex subsets. The classifier was parameterized using 5 times cross-validation on the train co-complex interactions and applied to 17.5 million pairs of proteins, scoring each one for their ability to interact based on the input data. See materials and methods. The full list of scored pairs and the complete feature matrix can be found in the data availability section. To evaluate the performance of our SVM classifier, we examined the confidence scores of 8,337 co-complex interactions in our leave-out test set using a precision recall framework, Fig 2a. Precision recall frameworks are the preferred method of evaluating imbalanced binary classification problems such as protein interaction identification. Davis and Godrich, 2006, Saito and Riemsmeyer, 2015. We see that Hu.MAP 2.0 outperforms our previous complex map, Hu.MAP 1.0, as well as all other datasets including Bioplex 2, Wen et al. And Hein et al., demonstrating the power of data integration. For example, a direct comparison of Hu.MAP 2.0 to Hu.MAP 1.0 at a fixed precision of 0.5 results in an increase of 67% recall relative to Hu.MAP 1.0. We also evaluated the increase in total high confidence, precision equals 0.9, protein interactions identified by Hu.MAP 2.0 and saw an increase of greater than 1,880 high-confidence interactions over Hu.MAP 1.0. Additionally, we see a drop in performance when we remove the WMM features from Hu.MAP 2.0, further showing how important the WMM features are to performance improvements. Finally, we compare Hu.MAP 2.0 to the Hu-Re dataset, which encompasses a 17,500 x 17,500 all-by-all yeast-2 hybrid screen, Luck et al., 2020. Yeast-2 hybrid aims to capture only direct protein-protein interactions and has previously shown good performance on benchmarks of binary interactors, Luck et al., 2020. Here we see Hu-Re underperforms all other networks when evaluated on co-complex interactions, likely due to its inability to identify indirect physical interactions. An external file that holds a picture, illustration, etc. object name is MSP17E10016G005.jpg. Figure 2. Hu.MAP 2.0 outperforms previous complex maps. Precision recall, PR, plot evaluated on a test, leave out, set of literature curated co-complex pairwise protein interactions. The plot shows Hu.MAP 2.0 is more accurate and comprehensive than previous published datasets. The plot also evaluates the performance of predictions without the weighted matrix model, WMM, and shows the WMM substantially improves performance. Clustering workflow used to identify protein complexes in Hu.MAP 2.0 protein interaction network. The network is first filtered based on the confidence score produced by the support vector machine, SVM. The filtered network is then clustered using a two-stage approach, clustering first using cluster 1, and then further clustering with MCL. The resulting clusters are then evaluated using the K-clique method, see materials and methods, on training complexes. Approximately 1,700 parameter combinations were evaluated, each producing a unique set of clusters, sweeping SVM score filter thresholds, and clustering parameters, i.e., cluster 1 max overlap, cluster 1 density, and MCL inflation. K-clique precision recall, KPR, scatterplot of 1,700 clustering parameter sets. 5 clusterings, colored hollow circles, were selected representing varying degrees of confidence, balancing the trade-off between precision and recall. The 5 selected clusterings were combined as a final set of clusters, orange-filled circle. KPR scatter plot of Hu.MAP 2.0 complexes, orange-filled circle, and other published complex maps, colored-filled circles, evaluated on a test set of literature-curated complexes. Hu.MAP 2.0 complexes increase in both precision and recall relative to other maps. Also plotted are the 5 sets of complexes at different levels of confidence, colored hollow circles, demonstrating consistency between the level of confidence determined from training set, B, and test set. Recently, a co-regulation map based on protein expression was shown to capture relationships among proteins that do not necessarily interact or co-localize, Kostacher et al., 2019. This dataset therefore provides an independent test of the quality of our protein interactions. When we compared the Hu.MAP 2.0 interactions to the most co-expressing pairs in Kostacher et al., we see a highly significant overlap, p-value less than 10e-10, hypergeometric test, indicating a high degree of consistency between the orthogonal datasets. Hu.MAP 2.0 complex map. Once we confirmed our protein interaction network was of high quality, we then clustered this network using a parameterized two-stage clustering algorithm to identify highly connected proteins which represent protein complexes. Briefly, the algorithm first filters the network based on the confidence score from the SVM classifier and then uses the cluster 1 algorithm, Napus et al., 2012, to cluster the thresholded network. Cluster 1 often produces large clusters containing proteins from multiple complexes. To remedy this, each resulting cluster is further clustered using the MCL algorithm, Enright et al., 2002, Fig 2b. There are several parameters for each algorithm that require optimization in addition to an SVM confidence score threshold of the input network. We therefore take an agnostic approach and optimize these parameters by generating clusterings for over 1,700 parameter combinations. Each parameter combination is evaluated using the K-clique precision recall performance measure, Drew et al., 2017, comparing the resulting clusters from the specific parameter combination with the quorum-based set of training complexes, Fig 2c. The resulting clusterings vary substantially with regards to their performance but ultimately show a familiar pattern of a trade-off between precision and recall. We therefore selected 5 clusterings that balance the precision recall trade-off. For example, clustering 1, green, is a set of clusters with extremely high precision but low recall, while clustering 5, gray, is a set of clusters with medium precision yet higher recall, Figs 1a and N2c. 2c. This allows us to rank our confidence of a protein complex based on its appearance in one of the 5 clusterings. We then combined all 5 selected clusterings into a union set while preserving their precision rank, dataset EV1. To evaluate the union of clusterings that represent our final Hu.MAP complexes, we again use the K-clique precision recall performance measure but now calculated on the leave-out set of test complexes. As shown in Fig 2d, our Hu.MAP 2.0 complexes balance both precision and recall. Additionally, we see a consistent trend of K-clique precision and recall values for our individual clusterings between both the train, Fig 2c, and test sets, Fig 2d. This suggests the confidence ranking given to each complex is robust. We also observe our final set of complexes outperforms our previous Hu.MAP 1.0 complexes and other previous state-of-the-art complex maps. Taken together, this points to Hu.MAP 2.0 complexes as being highly accurate and spanning a large portion of all human protein assemblies. Identification of multifunctional promiscuous proteins. Now that we have established Hu.MAP 2.0 as a highly accurate resource of human protein complexes, we can ask questions about protein assemblies in a systematic way. Specifically, one systematic question we can ask is how prevalent are promiscuous proteins in stable protein assemblies? That is, how often do we see proteins participating in multiple different complexes and presumably performing orthogonal functions, sometimes termed moonlighting, Chappell et al., 2015, Jeffrey, 2015. We therefore created a non-redundant set of complexes, see materials and methods, and surprisingly identified 253 proteins that participate in multiple complexes, dataset EV2. This is a 53% increase over the same analysis if done on Hu.MAP 1.0. The majority of promiscuous proteins appear only in 2 complexes, and the largest number of complexes a protein participates in is 4, suggesting these are not just sticky interactions but rather proteins participating in multiple specific complexes. The 253 proteins constituted nearly 7.5% of proteins in the non-redundant set of complexes. This compares to 106 moonlighting human proteins identified in the MoonProt database, Mani et al., 2015. Additionally, when we compare expression levels from the Human Protein Atlas, Ullin et al., 2015, across tissues of promiscuous proteins versus non-promiscuous proteins, we see the bulk of the distribution substantially overlaps, suggesting the promiscuous proteins do not appear in a greater number of complexes due to higher expression levels, Fig. EV1. An external file that holds a picture, illustration, etc. object name is MSB17E10016G008.jpg. Figure EV1. Density plot of Human Protein Atlas, Ullin et al., 2015, transcript expression across tissues of promiscuous proteins versus non-promiscuous proteins. We observe negligible differences between promiscuous and non-promiscuous distributions, suggesting expression levels are not a factor contributing to the identification of promiscuous proteins. Figure 3a and b shows an example of a promiscuous protein, HSPA9, participating in 2 unrelated protein complexes. HSPA9 is a multilocational, mitochondria and nucleus, and multifunctional protein, playing a role in mitochondrial import and stress response, Wadhwa et al., 2002. We identify HSPA9 participating in 2 complexes that reflect its multifunctional role. First, we identify HSPA9 in complex Hu.MAP 2_01130, a heat shock response complex supported by APMS experiments, Guruharsha et al., 2011, Hutlin et al., 2015, and cofractionation evidence, Wen et al., 2015. And second, we identify HSPA9 in complex Hu.MAP 2_00358, a mitochondrial protein import complex supported by APMS experiments, Malavanaya et al., 2011, Hutlin et al., 2015, Bolt et al., 2016, cofractionation evidence, Wen et al., 2015, and WMM based on, Triber et al., 2017. To further verify HSPA9's membership in these 2 complexes, we inspected sparkline traces of 2 cofractionation experiments, Wen et al., 2015, Fig. 3c. We identified 2 separate elution peaks of HSPA9 which correspond to the 2 complexes. This example shows the ability of our complex map to identify multifunctional promiscuous proteins and place them into their respective non-overlapping functional complexes. An external file that holds a picture, illustration, etc. object name is MSB17E10016G006.jpg. Figure 3. Hu.MAP 2.0 complexes identify promiscuous proteins. A. B. Multifunctional protein HSPA9 participates in 2 distinct complexes, Hu.MAP 2_00358, A, and Hu.MAP 2_01130, B. Hu.MAP 2_00358, turquoise, high confidence, is enriched for reactome annotation mitochondrial protein import, a known function of HSPA9. Hu.MAP 2_01130, blue, very high confidence, is enriched for reactome annotation regulation of HSF1-mediated heat shock response, another known function of HSPA9. Weight of network edges represent confidence of interactions. C. Sparkline elution profiles from 2 orthogonal biochemical fractionation experiments. HEC 293 cell lysate was separated using a mixed bed ion exchange column, and Drosophila melanogaster embryo lysate was separated using a heparin column, Wen et al., 2015. HSBA9 elutes in two distinct peaks, shaded, which co-elute with members of the two complexes. X-axis represents fraction collected along biochemical separation. Y-axis for each row represents observed protein abundance. D. Promiscuous proteins are older on average than single complex proteins. Z-scores for each protein age group were determined by comparing the number of promiscuous proteins to a randomly sampled background set consisting of non-promiscuous proteins, i.e., participating in only one complex. Saeed and Dean, 2006, previously showed a strong relationship between protein age and interaction connectivity. We therefore hypothesize that the promiscuous proteins would be on average older due to younger proteins not having enough evolutionary time to make multiple connections. Figure 3D shows a clear enrichment in older proteins in the set of promiscuous proteins and a depletion for younger proteins. See materials and methods. Using G-Profiler, Riamond et al., 2016, to identify functionally annotations for the older promiscuous proteins, we identify older promiscuous proteins are enriched for reactome metabolism, adjusted p-value 5 × 10^-8, GSCS corrected, among other metabolism-related annotations, Fig EV2. Consistent with this finding, many moonlighting proteins are enzymes with multifunctional roles, Jeffrey, 2015. Our results suggest a protein's complex membership may play a role in its multifunctional activity. An external file that holds a picture, illustration, etc. object name is MSB17E10016G001.jpg. Figure EV2. Annotation enrichment of older promiscuous proteins. G-Profiler output shows older promiscuous proteins are enriched for metabolic processing annotations. Functional annotation of uncharacterized proteins. High-quality protein complex maps have long been sought after for the purpose of functionally annotating poorly characterized proteins in a genome due to the relationship between physical interaction and biological function, Gavin et al., 2002, Ho et al., 2002, Wang and Marcotte, 2010. To assess Hu.MAP 2.0's ability to functionally annotate uncharacterized proteins, we first tested whether our identified complexes are enriched with literature-curated annotations including gene ontology, Ashburner et al., 2000, Reactome, Fabregat et al., 2016, Quorum, Giurgiu et al., 2019, Human Phenotype Ontology, Kuhler et al., 2014, and Keg, Kanaiza et al., 2014. We see in Fig EV3 that greater than 40% of our complexes are enriched with at least one annotation, which is 20.5-fold higher than expected by randomly shuffled complexes. This result shows Hu.MAP 2.0 complexes are functionally coherent. An external file that holds a picture, illustration, etc. object name is MSB17E10016G004.jpg. Figure EV3. Hu.MAP 2.0 complexes are functionally enriched. The bar chart shows the number of identified complexes that are enriched with at least one annotation from Go, Reactome, Quorum, Keg, or Human Phenotype Ontology, HP, at an FDR threshold of 0.05. As an example of the utility of Hu.MAP 2.0 in annotating poorly characterized proteins, Fig 4a shows two previously unreported interactions with Rene2, CMTR1, and SET3. The evidence for these interactions is elucidated from cofractionation mass spectrometry experiments which demonstrate a high degree of correlation over multiple separation column types and multiple organisms, which we have shown suggests a deep conservation in function, Wen et al., 2015, Fig 4b. Rene2 is implicated in Aicardi-Goutier's syndrome, a monogenic autoinflammatory disorder which mimics in utero viral infection of the brain, Rianes and Jackson, 2014. Mechanistically, Rene2 degrades RNA fragments of RNA-DNA hybrids including Okazaki fragment RNA primers during DNA replication, Chapados et al., 2001. Mutations in RNA2 are thought to disrupt the degradation of immunostimulating nucleic acids and cause innate immune activation. Riens and Jackson, 2014. CMTR1 is a mRNA methyltransferase and a known regulator of protein expression of IFN-stimulated genes to restrict viral infection. Williams et al., 2020. CET3, also a methyltransferase, is a human host protein critical for infection of a wide range of viruses and participates in viral replication, yet its mode of action is currently unknown. Deep et al., 2019. The links we identify between CMTR1, CET3, and RNA2 point to the hypothesis where CMTR1 and CET3 interact with RNA2 to modulate the innate immune response and affect viral replication. An external file that holds a picture, illustration, etc. object name is MSP17E10016G009.jpg. Figure 4. Transfer of function annotations to uncharacterized proteins. CET3 and CMTR1 are identified as co-complex interactors with the ribonuclease H2 complex, which provides a possible mechanistic explanation for their role in viral infection. Sparkline elution profiles from multiple orthogonal co-fractionation experiments demonstrate a strong degree of co-elution among subunits in the CET3 CMTR1 RN OSA H2 complex. Weight of network edges represents confidence of interactions. X-axis represents fraction collected along biochemical separation. Y-axis for each row represents observed protein abundance. The uncharacterized protein, C7orf26, is identified as part of the integrator complex. Sparkline elution profiles show a high degree of correlation between C7orf26 and subunits of the integrator complex from multiple orthogonal co-fractionation experiments. The association of C7orf26 and integrator complex is additionally supported by affinity purification mass spectrometry, APMS, experiment where C7orf26 is pulled down with integrator subunit dates. The uncharacterized protein, CCDC9, is identified as co-complex with the exon-exon junction complex, EJC, a ribonucleoprotein complex involved in splicing. Sparkline elution profiles from the independently collected RNA diffract size exclusion chromatography, SEC, experiments show CCDC9 co-elutes with known subunits of the EJC when RNA is present, black. The elution profiles also show CCDC9 is sensitive to RN-OSA treatment, shift of elution peak between black and red profiles, as are the subunits of the EJC, further supporting CCDC9's participation in this known ribonucleoprotein complex. It has been observed that biomedical research is biased toward the study of well-annotated genes, and this bias is due less to the physiological importance or disease relevance of the gene but rather to the ease of experimentation using traditional methods. Stoger et al., 2018. Unbiased systematic approaches such as the integration of thousands of mass spectrometry experiments described here provide a powerful tool for closing the gap of uncharacterized proteins. We therefore cross-referenced genes deemed poorly annotated, UniProt, the UniProt Consortium, 2017, annotation score less than or equal to 3, with Hu.MAP 2.0 complexes that were enriched for functional annotations. We identified 274 proteins in which we could transfer the annotation of the complex to an uncharacterized protein, dataset EV3. This is an increase of 40% over the same analysis done with Hu.MAP 1.0. Within this set of uncharacterized proteins, we identify C7orf26 as a member of the integrator complex, Fig 4c. We see strong correlation of C7orf26 with integrator subunits in co-fractionation experiments from both HEC293 cells and fly embryos, Fig 4d. In addition, C7orf26 was also identified in several separate affinity purification experiments, Malavania et al., 2011, where integrator subunits were the target bait protein, Fig 4e. The consistent results from these orthogonal datasets suggest a role for C7orf26 in the integrator complex and provide a new function for a completely unannotated protein. We also observe another uncharacterized protein, CCDC9, as being a member of the exon-exon junction complex, EJC, Fig4F. The interaction between CCDC9 and the EJC is supported by evidence from Hein et al. 2015, 8PMS experiments and WMM evidence. Since the EJC is a ribonucleoprotein complex involved in RNA splicing, we searched our previously collected RNA diffract mass spectrometry data, Malum et al., 2019, for evidence of CCDC9 as being both a member of the EJC and also associated with RNA. The diffract experiment identifies ribonucleoprotein complexes by comparing elution profiles of protein complexes with and without RNA treatment. We see in Fig4G CCDC9 not only co-elutes with EJC subunits but is also sensitive to RNA treatment, suggesting it is interacting with the EJC while associated with RNA. Importantly, the diffract experimental data were not included in the generation of Hu.MAP 2.0 and therefore represent an independent assessment of CCDC9's interaction with the EJC. Additionally, CCDC9 was identified as an RNA-binding protein in high-throughput screens searching for mRNA-binding proteins, Bouts et al., 2012, Costello et al., 2012, which is consistent with our observation of CCDC9 participating in a ribonucleoprotein complex. Go to Discussion. Herein, we describe the construction of the most accurate and comprehensive protein complex map to date, which fills a large gap in our knowledge regarding the composition of functional complexes in the cell. We demonstrate the utility of our map by assigning functions for hundreds of completely uncharacterized proteins, providing testable hypotheses for their characterization. Additionally, we determine the prevalence of proteins that participate in multiple independent protein assemblies, including ones with disparate functions, suggesting moonlighting functions for the protein. Overall, our results, searchable with a simple web interface, http://umap2.proteincomplexes.org/ establish the utility of Hu.MAP 2.0 for furthering our understanding of human protein functions. Summary of methodological updates to the Hu.MAP pipeline from version 1.0. While the substantial performance gain between Hu.MAP 1.0 and Hu.MAP 2.0 is largely due to the addition of newly published interactome datasets, we summarize changes between the pipelines here. First, the Hu.MAP 1.0 pipeline took advantage of Libsum's, Chang and Lin, 2011, grid.py utility to sweep SVM hyperparameter C and gamma. The grid.py utility relies on the accuracy metric for evaluation defined as accuracy equals TP plus TN, TP plus TN plus FP plus FN, where TP is true positives, TN is true negatives, FP is false positives, and Fn is false negatives. Using accuracy as an evaluation metric is problematic in cases of large class imbalance such as protein interaction prediction. Specifically, there are orders of magnitude more negative interactions than positive interactions, and therefore, accuracy can be dominated by the true negatives, resulting in a poor predictor of new interactions. In the Hu.MAP 2.0 pipeline, we therefore remedied this by developing our own cross-validation utility, https://github.com/marketLab/protein_complex_maps/tree/master/protein_complex_maps/model_fitting/cross_validation, which uses area under the precision recall curve, PREUC, as a metric to identify optimal C and gamma SVM parameters. PREUC does not rely on true negatives for its calculation and is preferred for evaluation of problems with large class imbalance, Davis and Godrich, 2006. Second, we simplified the clustering section of the Hu.MAP 2.0 pipeline to use only cluster 1 followed by MCL. In the Hu.MAP 1.0 pipeline, we observed a gain in performance when we combined results from the Newman clustering method, Newman, 2004, with the MCL results. In the Hu.MAP 2.0 pipeline, we do not see such gains and opt for only using MCL. Third, the Hu.MAP 1.0 pipeline did not classify protein complexes by their estimated confidence level. Hu.MAP 2.0 now allows for a quick way for users to evaluate a complex of interest based on a confidence measure. As described above, in the Hu.MAP 2.0 pipeline, we use the K-clique precision recall performance measure for evaluating the generated clusterings. The K-clique precision recall measure allows for a finer grain analysis of performance due to the separation of precision in recall terms as opposed to the F-grand K-clique measure used in Hu.MAP 1.0. We were then able to select 5 clusterings that provided a trade-off between precision and recall, which ultimately serve as a confidence score for the complexes in those clusterings. Hu.MAP 2.0 identifies complexes across a broad distribution of biochemical classifications. A primary goal of our work is to identify all stable physical complexes in human cells. While Hu.MAP 2.0 substantially increases coverage of stable human complexes, the results ultimately reflect the integrated high-throughput method's ability to identify these assemblies. For example, high-throughput biochemical purification techniques such as APMS and CFMS are commonly thought to be robust at identifying soluble cytoplasmic protein complexes and less robust at purifying, for example, membrane-bound complexes. To determine whether our Hu.MAP 2.0 complexes were biased against certain categories of proteins, we used PantherDB, Me et al., 2019. Go analysis to identify enriched and depleted terms. Interestingly, we see Go cellular component terms such as vesicle membrane and mitochondrial membrane as enriched with 1.27 and 1.41-fold enrichment respectively. Alternatively, we see a depletion in proteins annotated with plasma membrane in Hu.MAP 2.0 yet only slightly, with a 0.86-fold depletion and a total of over 2,300 proteins with the plasma membrane annotation in our complexes. To further investigate the validity of these proteins in our data, we searched for examples of plasma membrane complexes. We found several high-confidence complexes enriched with plasma membrane term including a potassium voltage-gated channel complex, Hu.MAP 2_0584, and a fibroblast growth factor receptor complex, Hu.MAP 2_06274. This suggests that while the high-throughput methods we integrate in our pipeline may have some biases against membrane-associated proteins, our map still has substantial coverage of these proteins and produces coherent and biologically meaningful complexes. To further explore biases in our dataset, we considered the degree to which our complexes are distributed across all tissues/cell types or are they cell type specific. To evaluate this potential bias, we considered transcriptomics data in the Human Protein Atlas, across 37 tissues, 43 single cell types, 10 main regions of each mammalian brain, 18 blood cell types, and 69 cell lines, Woolen et al., 2015, as an indicator for protein expression. We evaluated these data for the entire human proteome, Fig 5a, proteins identified in Hu.MAP 1.0, Fig 5b, and those identified in Hu.MAP 2.0, Fig 5c. In the entire human proteome, we see a substantial number of proteins likely expressed in all samples, 50%, but still many displaying some specificity. In Hu.MAP 1.0 and Hu.MAP 2.0, we see the vast majority of proteins likely expressed in all samples, 76 and 70%, respectively. This suggests that the complexes we identify are likely to be expressed across many different cellular conditions and cell types. As more and more high-throughput datasets are collected on different cell types and tissues, we expect to identify more cell type-specific complexes upon their integration. An external file that holds a picture, illustration, etc. object name is MSP17E10016G011.jpg. Figure 5. Protein complex map coverage across human protein atlas tissues and cell specificity. Coverage of all human proteins shows a broad distribution of proteins classified into a range of specificity classes, from detected in all tissues and cells to detected in only a single tissue or cell type. Coverage of Hu.MAP 1.0 proteins show a narrower distribution of proteins classified into specificity classes, with the majority of proteins detected in many or all tissues and cell types. This suggests Hu.MAP 1.0 represented the core cellular machinery. Coverage of Hu.MAP 2.0 proteins show a distribution representative of the core cellular machinery shared among all or many tissue and cell types, but also shows an increase in cell type specificity with gains in proteins that are only detected in some tissue/cell types. Completeness of human protein complex map. Our goal in this work is to build a complete and accurate set of protein complexes. We next asked how far have we come in achieving this goal? The size of the human interactome has previously been estimated to contain 154K 369K total interactions, Hart et al., 2006. Here we report 57K distinct interactions equating to roughly 15 to 37% complete. Consistent with this, if we consider the quorum benchmark we use for evaluation in Fig 2a as representative of all interactions, we see the majority of the Hu.MAP 2.0 precision recall curve in Fig 2a roughly falls between 15 and 37% recall. Also, making the same assumption for the quorum benchmark, we can get rough estimates of Hu.MAP 2.0's coverage of total protein complexes. The precision and recall metrics used in Fig 2d are robust to any redundancy and therefore give an accurate representation of total coverage. In Fig 2d, we see Hu.MAP 2.0 covers greater than 30% of complexes in the benchmark at a precision of 60%. As described in the section above, many cell type and condition-specific interactions likely make up a large portion of the remaining undiscovered interactions. We expect these interactions to be a focus of future experimentation in order to gain greater coverage of the complete human interactome. Go to Materials and Methods. Reagents and Tools Table. Reagents/resource reference or source identifier or catalog number. Software. MCL version 14 to 137 https://mycons.org/mcl/Enright et al., 2002. Cluster 1 version 1.0 https://pacanerolab.org/cluster1/Napus et al., 2012. libsvm version 321
https://www.chi.nshu.edu.tw/tildekun/libsm/Chang and Lin, 2011. Upset version 0.4.1 https://upsetplot.rethidocs.io/n/stable/Lex et al., 2014. MS Blender version EC3B484 https://github.com/marketlab/msblender, Quan et al., 2011. Protein Complex Maps version 512b9b5 https://github.com/marketlab/protein_complex_maps, Drew et al., 2017. Open in a separate window. Methods and Protocols. Mass spectrometry dataset collection. Mass spectrometry data and features based on those data used as input into the machine learning classifier were collected from various publications. Specifically, protein interaction features for datasets used in Hu.MAP 1.0, Drew et al., 2017, for example, Wen et al., Hind et al., Hutlin et al., were downloaded from http://hu1.protein-complex.org/static/downloads/feature_matrix.txt.gz. 4 vector comparison measures were used for cofractionation data from Wen et al., including Poisson noise Pearson correlation coefficient, weighted cross correlation, co-apex score, and MS1 ion intensity distance metric.
All 4 vector comparison measures were applied to each of the 55 fractionation experiments, totaling 220 features. Pairs of proteins were filtered to ensure co-fractionation measures were more than 0.5 in at least 2 species. APMS data from Guruharsha et al., 2011, mapped onto human orthologs using in Paranoid, Sunhammer and Erstland, 2015, were represented using the score value originally downloaded from supplemental table S3 and Guruharsha et al. APMS data from Malavanaya et al., 2011, were represented by the memos, core modules, certainty assignments approved, provisional, and temporary originally downloaded from supplemental file S1, assigning the scores 10, 3, and 1, respectively. Bioplex 1.0, Hutland et al., 2015, features were used as originally downloaded from http://ren.hms.harvard.edu/bioplex/data/cdf/150408_var2594.cdf including NWD score, z-score, plate z-score, entropy, unique peptide bins, ratio, total PSMs, ratio total PSMs, and unique total peptide ratio. For the Hein APMS data, the features pray.bait.correlation, valid.values, log10.pray.bait.ratio, and log10.pray.bait.expression.ratio were taken from supplemental table S2 in Hein et al., 2015. The mean value was used across the experiments in the case of multiple entries for a given protein pair. Note, all human net, Lee et al., 2011, features were excluded from all model training. New datasets added for Hu.MAP 2.0 were downloaded from original publications or associated dataset web resources as shown in Table 1. The same measures used for Bioplex 1.0 were also used for Bioplex 2.0 features specifically NWD score, z-score, plate z-score, entropy, unique peptide bins, ratio, total PSMs, ratio total PSMs, unique total peptide ratio and average assembled peptide spectral matches. We used the measures from the proximity labeling dataset, Gupta et al., for both the ciliated condition and non-ciliated condition, specifically average spectra, average Saint probability, max Saint probability, fold change, and Bayesian FDR estimate. The same measures were used for the proximity labeling Yoon et al. Data but only for the single condition. Measures used for Bolt et al. Data were socio-affinity index, SIGE, and individual terms based on the spoke model for where protein I is the bait, SHIJ, and where protein J is the bait, SHI. The matrix model term for the socio-affinity index was also used, MAE. Mass spectrometry data from Triber et al., 2017, were downloaded from the PRIDE web resource, Perez-Riverol et al., 2019, PXD004193, and reprocessed using the MS Blender pipeline, Kwon et al., 2011. Full details are described in Malum et al., 2019. Only WMM features, described below, were calculated for Triber et al. Table 1. Integrated mass spectrometry datasets. Dataset Num of experiments link. Wen et al., 2015. 5344 fractions http://hu1.protein-complexes.org/static/downloads/feature_matrix.text.gz. Hein et al., 2015. 1125 bait pull downs http://hu1.protein-complexes.org/static/downloads/feature_matrix.text.gz. Bioplex 2.0, Hutland et al., 2017. Includes Bioplex 1.0. 5891 bait pull-downs https://bioplex.hms.harvard.edu/data/bait prepares_no filters_bp2a.tsv. Gupta et al., 2015. 2 conditions x58 proximity labeled baits equals 116 http://prohitsweb.lunenfeld.ca. Yoon et al., 2018, 119 proximity labeled baits http://www.cell.com/cms/2118963855/2087347233/mmc2.xlsx. Bolt et al., 2016. 217 bait pull-downs https://static-content.springer.com/ism/art%3a10.1038%2fmcoms11491/media objects/41467_2016_measm835_esm. xlsx. Triber et al., 2017, reprocessed by Malin et al., 2019, 3004 RNA hairpin pull-down MS runs https://www.cell.com/cms/10.1016/j.cellrep.2019.09.06//attachment/45ab-EFF47528906DC5E1184807 Open in a separate window. Hu.MAP dataset, Luck et al., 2020, which was not included in training but was included for evaluation, was downloaded from http://interactome.batterlab.org/data/hu.re.tsv. Hu.re protein interactions were ranked based on the number of assays the interaction was identified in. Weighted matrix model. To gain additional information on the probability that two proteins interact, we generated additional features using a WMM. The WMM is based on the hypergeometric distribution and is described in Hart et al., 2007, and Drew et al., 2017. Briefly, we used the hypergeometric test, equation 1, where K represents the number of experiments when both proteins A and B are identified. Variables N and M represent the number of experiments that independently identified protein A and protein B respectively. N represents the total number of experiments. The index I is defined from K to the minimum of N and M. P #shared experiments greater than or equal talk vertical bar N, M, N equals I equals Kmin N. M Ni N and MINM. 1. Our implementation of the WMM is based on presence or absence of proteins in individual experiments, for example, one pull down. Due to the nature of high-throughput experiments, noise arises in the form of spurious identifications leading to a protein being erroneously called present in the experiment. To deal with this noise, we set arbitrary but sensible cutoffs of the quality of identification required for a protein to be considered present in the experiment. WMM for Bioplex 2.0 was calculated only considering experiments for a given protein where the protein had greater than 2.0 Bioplex 2.0 z-score and greater than 4.0 Bioplex 2.0 z-score. WMM based on Gupta et al. Were calculated considering all experiments, greater than 2 average spectral counts, and greater than 4 average spectral counts. WMM based on Bolt et al. Were calculated considering all experiments in greater than 4 spectral counts. WMM based on Triber et al. Were calculated considering greater than 2 spectral counts in greater than 4 spectral counts. WMM based on Yoon et al. Were calculated considering all experiments, greater than 2 spectral counts in greater than 4 spectral counts. For each calculation, we generate a feature in the form of the negative natural log p-value of equation 1, and the total number of experiments the pair of proteins is observed together, i.e., pair count. All features, pre-calculated from original publications and WMM, were combined into 17,564,755 protein pairs times 292 features matrix. Since not all features cover all protein pairs, missing values were filled with 0.0. The final feature matrix can be found in the data availability section. Gold standard test and training set. To create a test and training set of literature curated protein complexes, we downloaded the complete set of quorum complexes, Georgiou et al., 2019, version 2017_07_02, http://mips.helmholtz-munchen.de/quorum/download/quorum_2017_07_02.zip, and filtered out all non-human proteins. Complexes were merged to eliminate redundancy, so no two complexes had greater than 0.6 Jacquard coefficient. Complexes were then randomly split into test and training sets. A complex was removed from the test or training sets if any pairs of proteins overlapped in the other set. Large complexes greater than 30 subunits were removed from the test and training complexes. Test and training sets were also generated for pairs of proteins for training the SVM classifier. A pair of proteins was labeled positive if both proteins were in the same complex. A pair was labeled negative if proteins were in separate complexes. All other pairs were left unlabeled. For test and training pairs, only 10% of pairs from large complexes were considered. Below is the command line used to generate the test and training sets.
./protein_complex_maps/preprocessing_complexes/split_complexes.py input_complexes all complexes_20170702_geniads_human.text random_seed 1234 size_threshold 30 subsample_large_complexes 0.1 remove_large_complexes remove_largest merge_threshold 0.6. Additionally, the full test and training sets used in this study can be found in the data availability section. Support vector machine model selection and evaluation. We trained a SVM classifier using Libsm, Chang and Lin, 2011, to classify pairs of proteins as co-complex protein interactions. We first generated a feature matrix using the features described above, where rows are pairs of proteins and columns are features. The feature matrix was further labeled using the gold standard training set described above. We used five-fold cross-validation using only the training set when training to select SVM parameters C and gamma. We evaluated a range of C values, 2, 8, 32, 128, 512, and gamma values, 0.000488281225, 0.0019531225, 0.0078125, 0.03125. As an evaluation metric, we used area under the precision recall curve, ALPRC, averaging across the 5 cross-validation sets. We identified C equals 512 and gamma equals 0019531225 with the highest cross-validated ALPRC. We retrained a full model using all training data using these parameters. We used this final model to predict on all pairs in the feature matrix. The final result is a list of pairs with a corresponding score generated by model. We evaluated the final model using a precision recall framework as shown in Fig 2a. We used the scikit-learn Python package, preprint, Buetink et al., 2013, to calculate precision in recall for the leave-out gold standard test protein pairs. For comparisons between datasets as shown in Fig 2a, we generated additional models restricting the features to just those generated from the given dataset, keeping the parameter C and gamma fixed. Note, the Hu.MAP dataset was evaluated using the dataset directly as described above. We additionally evaluated the SVM confidence score for its fidelity to the test set precision value. We observed that the test set precision is consistently higher than the confidence score,
Fig EV4. For example, a confidence score as low as 0.02 has 0.5 precision value. An external file that holds a picture, illustration, etc. object name is MSP17E10016G010.jpg. Figure EV4. SVM confidence score versus test set precision. The line plot shows the relationship between the SVM confidence score and the empirical precision value calculated from the test set of protein interactions. The relationship shows the precision value is consistently higher than the confidence score. Two-stage clustering and parameter set selection. We next used a two-stage clustering approach to identify clusters within the protein interaction network generated by the classification step described above, Fig 2b. First, the network was thresholded based on the SVM score. We then applied the Cluster 1, Napus et al., 2012, algorithm to identify dense regions in the thresholded network. Further, for each dense region produced by Cluster 1, we applied the MCL, Enright et al., 2002, algorithm to identify clusters. To identify optimal parameters for the score threshold, Cluster 1 parameters density and overlap as well as MCL inflation parameter, we generated clusters for various parameter combinations. Specifically, we evaluated a range of parameters, SVM score threshold, 1.0, 0.99, 0.97, 0.95, 0.9, 0.8, 0.7, 0.6, 0.5, 0.4, 0.3, 0.2, 0.1, 0.05, 0.01, 0.005, 0.001, 0.0005, 0.0001, 0.00005, 0.00001, Cluster 1 max overlap, 0.6, 0.7, 0.8, density, 0.1, 0.2, 0.3, 0.35, 0.4, and MCL inflation, 1.2, 2, 3, 4, 5, 7, 9, 11, 15. We also compared using an unweighted graph as input into Cluster 1 versus a weighted graph and observed the unweighted graph had superior performance. A weighted graph was used for the MCL stage clustering. We additionally applied a post-clustering filter that removed nodes from a cluster that lacked edges that scored greater than the SVM score threshold. To evaluate the clusterings as shown in Fig 2c and d, we used the K-Cleeks method, specifically weighted recall, R_weighted, and weighted precision, P_weighted, which we described previously, Drew et al., 2017. Briefly, the K-Cleeks method globally compares a set of clusters to a set of gold standard complexes by comparing cliques derived from the clusters to cliques derived from gold standard complexes. This comparison is done for all clique sizes from size 2, i.e., pairs, to size n, i.e., the size of the largest complex or cluster. A precision and recall value are calculated for all clique sizes. A weighted average is then calculated for both precision and recall across all clique sizes, weighted by the number of clusters with size greater than equals to the clique size. This is to limit the bias effect that larger clusters will have on the final precision or recall value. We evaluated all clusterings using the K-Cleeks method, comparing to the training set of gold standard complexes. We selected 5 clusterings that optimize the trade-off between precision and recall as shown in Fig 2c. These 5 clusterings were then combined into a union set. Table 2 shows the clustering parameters used for the selected clusterings. Table 2. Selected clusterings parameters. Clustering confidence score threshold Cluster 1 density Cluster 1 overlap MCL inflation. 1 extremely high 1.0, 0.4, 0.69. 2 very high 0.7, 0.4, 0.69. 3 high 0.5, 0.4, 0.74.
4 medium high 0.04 0.4 0.72. 5 medium 0.02 0.10 0.62. Open in a separate window. We finally evaluated the individual selected clusterings and the union of the selected clusterings using the K-clique method by comparing to the leave-out set of gold standard test complexes, Fig 2d. In addition, we compare to previously published complex maps from Wen et al., 2015, Bioplex 1.0, Hutlin et al., 2015, Bioplex 2.0, Hutlin et al., 2017, and our original Hu.MAP 1.0, Drew et al., 2017. Identification of promiscuous proteins. To identify proteins which participate in multiple complexes, we first determined a set of complexes with limited overlap. To determine the degree with which one complex overlaps another, we developed a subcomplex index, equation 2, defined as SC equals vertical bar AB vertical bar A vertical bar 2, where A and B are complexes, i.e., sets of proteins. The subcomplex index is related to the Jacquard index but is normalized by the size of a single complex rather than the size of the union of both complexes as is done in the Jacquard index. We then calculated the subcomplex index for every complex compared to all other complexes. We then generated a set of complexes with limited overlap by selecting complexes that had SC less than 0.5 to all other complexes. We then identified all proteins that participated in multiple complexes from this reduced set of complexes. Calculation of protein age enrichment for promiscuous proteins. Protein ages were mapped using mode age in the main_human.csv file from Liebeskind et al., 2016. Z-scores for each age group were determined by comparing the number of promiscuous proteins to a background distribution. The background distribution was calculated by counting the number of randomly sampled non-promiscuous proteins, i.e., proteins that participate in only one complex, in each age group. Annotation enrichment, tissue specificity, and overall expression. Annotation enrichment was calculated for GO, Reactome, Quorum, KEGG, and Human Phenotype Ontology, HP, terms using G-Profiler, Riamond et al., 2016, for each individual complex. All proteins observed in the 15,000 mass spectrometry experiments were used as the background set. Annotations inferred by electronic transfer were ignored. To evaluate annotation enrichment for all complexes, we first generated a set of shuffled complexes where proteinids were reassigned to new clusterids. This has the effect of keeping both the number of clusters and the size distribution of clusters the same as the final set of Hu.MAP 2.0 complexes. In addition, this also has the effect of keeping the distribution of complexes per protein constant with the final Hu.MAP 2.0 complexes as well. Annotation enrichment for the shuffled set of clusters was done as described above. Using this background annotation enrichment from all categories, we calculated a 0.05 false discovery rate threshold. We used the Human Protein Atlas, HPA, Ullin et al., 2015, to compare tissue specificity between the full human proteome, Hu.MAP 1.0, and Hu.MAP 2.0. RNA tissue distribution data were downloaded from https://www.proteinatlas.org/download/proteinatlas.tsv.zip and mapped to proteins through gene names. Comparison of overall expression levels between promiscuous and non-promiscuous proteins shown in Fig EV1 was done using HPA using the same proteinatlas.tsv.zip file described above. The median value was calculated for all tissue RNA columns for each individual promiscuous and non-promiscuous protein, and the resulting distribution was plotted. Go to Author contributions. K.E., JBW, and EMM conceived of the study. K.E. designed all experiments and analyzed data. K.E. wrote software including software pipeline and web resources. K.E., JBW, and EMM discussed and interpreted the results, and